أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين. Those people who are with me for the preparation of the SCPS examination part 1, like medicine and allied, and those people who want to acquire knowledge, although they have cleared out the FCPS part 2 examination, but they are lacking in knowledge. For example, if you are learning or studying about the pathology and about the diseases associated with the muscles, then you have got the basic knowledge of the muscles. As far as the anatomy is concerned, histology is concerned, as neurological aspects are concerned, etc. To gain full knowledge, you have to follow this path. Otherwise, you will be lacking always in your life. So in this fashion, I'm not a person of the basic subjects. I am the person of the clinical subjects. Basically, I was trained for the clinical skills since last many years, and focusing more on the physical findings in the patient and related with differential diagnosis and then clinical diagnosis, most probable clinical diagnosis associated afterwards, confirmed by the investigations. I was trained previously like this. And in this whole of my life, in the last 20-25 years, there are 2 or 3 supervisors I have faced, and they are about 100% clinical persons, and they have got a lot of services given to the nation, but cut down many of the investigations. Battery of investigations was not their practice. They are not owned by any institute. They develop their skills independently, and I salute to them. Afterwards, I have been retired from the Dow University of Health Sciences, and then afterwards I was, dumped down in the house. And somebody, for the 2 years or 3 years, because of the registrar of the Jinnah Sindh Medical University insisted that I should deliver the lectures to the students of the Jinnah Sindh Medical University, that is Sindh Medical College. I have delivered lectures 2 or 3 years for them. After that period, I was dumbed down in the house, and nobody is actually dare to entertain me, even I would say. And I started this channel to improve the poverty of the medical education, because those graduates who are passing MBBS, I don't think they can handle the patient confidently. Confidence, power of decision are always lacking in them. They are dependent upon the lot of investigations, and then they are able to diagnose, and after the diagnosis, even afterwards, they cannot manage the patient properly, in a proper fashion and recommended way. I started this channel because I can't go outside. Nobody is accepting me, no institute in this country. However, outside this country, there are 3 countries who offered me, but I negate them because I don't want to go to outside and benefit their students. I want to benefit my students. I started this channel, but however, I am very sorry that the people do not respond this channel, although I am delivering this channel and making and delivering lectures. And even those students who are affiliated with me in the past has gone. Only 1 or 2 or 3 students are left. So I am talking to the walls of my house, and those who are inside the wall, they are Jannat. I am talking to the Jannat now. because nobody is listening to these lectures. They don't want these lectures. They want short pathways. They want to pass this examination in any way, by hook or by crook. Okay, fine, no problem. I'm staying in this house or down in the house till my death, and afterwards I will be buried in the earth, in the grave. That's all. That is the end. That will be the end of the story of so-called Professor Sayyid Ali Haider, the facilitator, global honorary professor of medicine without any name or fame or economics. I do not want to take advantage from my people. I want to take mercy from the God, Allah Jalla Jalaluhu. That's all. I've cleared now the muscle. This is muscular system I'm talking about. There are a lot of things which have developed in the last 10 years, or 20 years, you can say. So skeletal system has been described by me, although I am not happy with my description also. So I have to go to very, very quick to deliver my lectures. And that is the hallmark of anything. You have allotted a time to deliver your lectures. Half hour, 45 minutes, that's all. Whether the people don't listen or we want to, if they are listening, the message is coming in one ear and they are given the exit to the other ear because of the development of the bilateral chronic otitis media. I don't want like this. So bit by bit I am talking about. If you are interested, listen to these lectures very carefully, calm and calm. Slowly and progressive. It is integrated, muscles which we are discussing because they are related. There are three muscles in the body. One is skeletal muscle, I have discussed. Second are the smooth muscles. The third one is the cardiac muscle. They are very, very, bound to each other. Integrated muscles. Now, integrated, what do you mean by integrated? You know that about it. What do you mean by integration? Means that in Urdu it is called inzamam. Inzamamul haq. Inzamam means that two or three things are colliding with each other. And in the simple words, in the dictionary it is written, integration means to form, coordinate, or blend into a functioning unit or unified as a whole, or to incorporate into a larger unit so that one or two, like Liaquat Ali Khan, मुक्का याद रखिएगा, उन्होंने कहा कि अगर मैं एक उंगली उठाऊंगा, इट मीन्स दैट ऑल राइट, अगर मैं पूरी उंगलियां दबाऊंगा तो एक मुक्का बन जाएगा, 5 सुबह होती है, और ये मुक्का अगर किसी की लग जाएगा तो उसको फाड़ खाएगा, उसके करीब नहीं आएगा. अगर यूनिटी रखते हैं आप, तो इंटेग्रेशन मीन यूनिट. So muscles, they are integrated. We are all formed from one line of a myelinic tissue that is mesenchyme. They are all muscles, whether they are skeletal muscle or smooth muscle or cardiac muscles. They are made of elongated cells we call myocytes, muscle cells, and muscle cell here in histology is called fibers. They have got nuclei, 1 nuclei, 2 nuclei, 3 nuclei, whatever that is. Skeletal muscle has been described as a typical examination, so it should be compared with the smooth muscles, or they should be compared with the cardiac muscle. Look this comparison. Skeletal muscle already mentioned is striated, they are voluntary, it's seen the striations, if you've seen the light microscope, in the bands are seen in the H bands and I band and so on, accordingly in the electron microscope. Looking at the smooth muscles, they are not striated, but on the light microscope they seem to be non-striated, and they are involuntary. Skeletal muscle under your control, and precentral gyrus, that is motor area. It's the order, it is the unit which is called the first order neurons, starting from there. The non-striated muscles are involuntary. They are usually associated with the autonomic nervous system. They are not under your control. in between the two, integration will cause what the cause is called the myocardium, and that is called cardiac muscles. They are stated like skeletal muscle, but they are involuntary, and they are functional units. Like this, it is present in the electron microscope like this. We'll discuss it, and under the light microscope you find this type of a constitution. Now this is the basic, most basic thing. You know and I know that. Skeletal muscles, the whole muscle is covered by the covering, and that is called the connective tissue covering, which is known as the epimysium, very thick, dense, irregular connective tissue. You know that about it. And it is called epimysium. And after, just in front of this covering, anteriorly or the whole of them, is covered by the fibrous tissue. You know that of the fibrous tissue, the superficial fascia and deep fascia. Deep fascia will causing the subtars to separate the different group of muscles. Afterwards, epimysium is the connective tissue covering of the muscle. Consists of the parallel skeletal muscle fibers, which are myocytes, arranged in the bundle separated by the connective tissue subtars. Inside, this is the subtars. They call them perimysium. Inside, longitudinally, it is divided into different units afterwards, a less thick layer of the dense irregular connective tissue. This is perimysium. And after the perimysium, it is the perimysium which are called the bundles are here, you see this. The individual fibers, now each fiber again is covered or separated by the other fibers by so-called the connective tissue layer, which is called endomysium. Now here I have already mentioned out previously why they are forming epimysium, why they are for perimysium, why they are forming the endomysium. I think you should understand. Previously I have told you. Here the light microscopic picture is seen is cylindrical in shape, non-branched. They are covered by the very should be non-branched. It's because they are a unit, a sarcomere. It is the functional unit, and multinucleated nuclei they are peripherally located close to the sarcolemma. Now, sarcolemma is the cell membrane. It is the cell membrane. Sarcoplasm, inside the cell there is a fluid cytoplasm of the muscle fiber is known as sarcoplasm. Now cytoplasm is acidophilic in nature and shows clear transfer station inside. Transfer station. This electron microscope will further elaborated that the sarcoplasm, now here starting with the sarcoplasm, which is not seen afterwards in the light microscope. You find the parallel myofibrils inside the sarcoplasm. Microfibrils. Inside the microfibrils, you find there are two types of fibers, they called myosin and actin. Now, numerous mitochondria are arranged in the rows between the myofibrils. Why they are mitochondria so much, and why they are a well-developed smooth endoplasmic reticulum, that is called the sarcoplasmic reticulum. Why? Because the myoglobin pigment, it is carry and store oxygen and glycogen, which is store the food. You need this mitochondria, you need the endoplasmic reticulum. The myofibrils of the skeletal muscles, now in deeply researched that the myofibrils are contractile threads or genales arranged longitudinally in the sarcoplasm. Each myofibril shows alternating dark A and the light band. The A band shows the pale area in the middle. This is the H band, which is divided by a dark line, M line. Seen here. Seen in the electron microscopy, the lines are present. Although they are not significant for you, importantly, but for the histopathology, if you want to study the diseases, that these lines are important. The A band shows the pale area in the middle, and the H band, which is divided by a dark line, which is M line. The I band, again, starting with the I band, shows a dark line This is the dark A, has been described. Now that becomes the light bands I. Shows a dark line in the middle, that is Z line, that is differentiated from that dark zone, dark bands. The sarcomere in the segment, sarcomere is the segment between the two successive Z lines. The sarcomere is concerned with the functional unit is the two successive Z lines. Z line has been described. Between the two Z lines there is what is known as sarcomere, is the contractile unit of the, what is the contractile unit of the myofibril? Is sarcomere? Yeah. It's a functional unit, it is the contractile unit. The myofibrils are, now coming to the myofibrils, are formed by the myofilaments. If you disintegrate them, it is thick myosin and thin actin filament. Myosin has got two hairs, it grasps the actin as already mentioned out. The A band is formed by the myosin. Here is the A band. A band forms the myosin myofilaments mainly, remember? And the terminal ends of the actin myofilaments. Terminal ends of the actin myofilaments. But the I band remaining is the I band. The I band is formed by the actin myofilaments. It's only the actin which participate in the formation of the I band. If you read one or twice, then you can understand with this the diagram is concerned. Now this is the deeper studies of the skeletal myofibrils. You study and study. There are a lot of things there are about the myofibrils. You know where the myofibrils. ऊपर से नीचे चले जाएं आप. शुरू करें डॉप फाशा, डीप फाशा जो था उसने सेप्टा दिए, उसने इंटर मस्कुलर सेप्टा दिए, उसके बाद अलग मसल ग्रुप के बना दिए, साइनेजेस, एगॉन वगैरह वगैरह फिक्सेटर. उन मसल्स को अब अलग अलग करके स्टडी कर रहे हैं तो उसके अंदर घुसते चले जा रहे हैं. अब इलेक्ट्रॉन माइक्रोस्कोप में हमने मायोफिब्रिल पहुंच गया, मायोफिब्रिल से अपने एक्टिन मायोसिन भी पाल दिया. अब एक्टिन और मायोसिन प्रोटीन हैं जिनके अपने स्ट्रक्चर्स हैं, स्पेसिफिक स्ट्रक्चर्स हैं, उनकी अपनी स्पेसिफिक प्रॉपर्टीज़ हैं. इसको अगर ध्यान में रखते हुए आप इसको मुवाज़ना कर लें, कंपेयरिंग विद द कार्डियक मसल इज फाउंड इन मायोकार्डियम स्टेटेड एंड इनवॉल्वमेंट. रिमेंबर इट इज अ syncytium means that the walls are not separating from each other. Although they are straight fibers which are seen as striation, but involuntary. Look like this. There are gaps, and if there is a stimulus here, it will spread like anything. You have to stop at some time, at some place where some fiber, some connective tissue membrane will stop it. Otherwise, it will enter into the other parts. Syncytium. This is a syncytium. Means that the walls, they are not completely separating these units, functional units. And the light microscope is cylindrical in shape, intermediate in the diameter, it is in between the smooth muscle and the skeletal muscle, the branch and stemose with each other. And covered by the thin, they are covered by the thin sarcolemma. They are mononucleated, although the other, you see that is the multinucleated in skeletal muscle, for example. Although they are very, very similar in the picture, the nuclei are oval shape and central, their nuclei are not oval shape. They are multinuclei and they are different shapes. And they are central, they are peripheral. Sarcoplasma is acidophilic and shows non-clear striation, fewer myofibrils. The sarcolemma in the skeletal muscle are very, very, consist a lot of myofibrils. Here you find shows non-clear striation. They are not very much number, higher in number. You do not need it here. Divided into the short segment cells, they are short segments divided, means cells, they are divided by the intercalated discs. The intercalated discs will be, this is the disc. This is the disc which is shown here. They are separated by the disc only. Now, electron microscope picture here, the few myofibrils are there, numerous mitochondria. You need always, because involuntary is a machine which will never stop in your life. Less abundant sarcoplasma reticulum, because the endoplasmic reticulum is the part where the proteins are assembled. You do not need much in in comparison with the skeletal muscle, glycogen and myoglobin means that they are storing this in the mitochondria. They are is the source provided the oxygen as well as the glycogen. They are the two food source, oxygen and glycogen. So the glycogen and myoglobin is present in the cells. Intercalated discs are the unique type of the disc. They are formed of the two cell membranes of two successive cardiac muscles. They are connected together by the junctional complexes, desmosomes and gap junction. The gap junctions are there here, only gap. So desmosomes are there, they are touching with each other, so one stimulus will be crossing like this. It's a syncytia. That's why I am calling syncytium, which is not present in the skeletal muscle, not never in the smooth muscle is concerned. It's a deep study which is going. Desmosomes are, is a structure in the two adjacent cells are attached, and the gap junctions allow the communication, the passage of the impulses between the two cardiac muscle cells, and that is why I am calling repeatedly and syncytia. Now comparing with this to the smooth muscle, because skeletal muscle and smooth muscle are two ends. Now between the two ends there is cardiac muscle. So light microscope picture of the smooth muscle, which are present in the whole body in the different parts in the vascular system and other connective tissue, they are fusiform in shape, spindle shaped cells. They are not longitudinal. The fibers. The small diameter in between the, if you comparing with the skeletal muscle, they are definitely smaller. Comparing with the other one, that is cardiac muscle, it is more than that. It is seen. Non-branched, they are not branching, but in cardiac muscle they are branched. But they are thin sarcolemma. I mean the cell membrane is thin. And mitochondria, the mononucleated cells are oval and central position. They're not in the peripheral position, the oval and central position, just like in the cardiac muscle. Sarcoplasm is non-striated. In the sarcoplasm, if you are doing electromyography, they are acidophilic but non-striated. They have the myofilament, no doubt, but they are not form striations. So electron microscopic picture is that the sarcoplasm now here contains mitochondria and sarcoplasmic reticulum. Abundant, not much like skeletal muscle, not much. They are cardiac muscle in between they are in the smooth muscle and in the skeletal muscle. And myosin and actin filaments inside it, if you are studying the myofibrils, they are irregularly arranged. Some is going transverse, some is going longitudinally, some is oblique. Irregularly arranged, that's why no striations, that's why the striations cannot be present in this. Although there is definitely striations are present, they are longitudinal fibers as far as skeletal muscle is concerned, as far as cardiac muscles are concerned. Cells are connected together by the gap junctions for the cell communication, so that one part of this is stimulated, the other parts can be stimulated, just like cardiac muscle. They are innervated by the autonomic fibers. Now, regeneration of the skeletal muscle cannot divide. Limited regeneration by the satellite cells, stem cells, if they are on the muscle cell surface, only limited. Cardiac muscle cell never, no generative activity. Smooth muscle cells can divide like uterus in the pregnancy. If they are destroyed, they can divide by meiotic division and mitotic and then form. And regenerate from pericytes, which is the stem cell of the smooth. The pericytes are the cells which have got, just like the action of a stem cell, it is a totipotent cell, which can differentiate it into the mesenchymal cell, and the mesenchymal cell will form the myo units, and myo units will form the smooth muscles. Active regenerative response can be achieved with the pericytes. The pericytes are active, it can be, divide, and it can be regenerate. Comparison can be done if you are site is skeletal muscle attached to the skeleton, myocardium of the heart, and viscera and smooth muscle vessels. Shape cylindrical, fusiform. Diameter largest, smallest, medium size. Branching, non-branched, and cardiac muscle is branched. Striations clear, it is not present in the smooth muscle, not clear because of the gap junctions, very, very higher sensitivity, it is not clear. Striations, although they are present. Intercalated discs are the unique things which is present in the cardiac muscle, not present in the skeletal and smooth muscles. Nuclei numerous and peripheral. In this case, if one central nucleus in a smooth muscle and one central nucleus in the cardiac muscle. Only central nucleus. Action is voluntary, and both cardiac and smooth muscle are involuntary. They are not ear controlled. Autonomic nervous system will supply. Regeneration is limited in the skeletal muscle, very difficult to achieve. And in the smooth muscle, active cardiac muscle, no. Hypertrophy should be there, no hyperplasia. Now this is the summary of the events which I have just told you before, that how this is the skeletal, the largest muscle, and the cardiac, medium-sized muscle, smooth muscles, or the smallest muscle, diameter is concerned, and this comparatively studies, in the cardiac muscle, the branching and the skeletal muscle is clear, just like this. I have already mentioned out in this tutorial that all types of the features. This muscular tissue, this is the muscular tissue which I have discussed. Skeletal, cardiac, smooth, their functions, their microscopic picture, whether it is the light microscope, whether it is the electron microscope. The functions are important. Remember what is called functional histology, how they are functioning. How they are functioning. This is another important point to be discussed afterward, the physiology. With how the myoneural junction, what happened there, I have already, that is why I have told you previously about the channels are concerned. That is chelonomics, which is a misnomer of most of us. We don't know about it. So this is the request from my side, that because of the people are not taking interest in my tutorials, because I am also a human being. You know that I am an old person, and I deliver these lectures for the sake of my people, for the students who are interested, whether they are interested or not. But the Jinnat is there for listening, no problem. They are the part of the world. They are listening to it, and they are admiring. I am 100% sure about it. I feel they are smiling and they are enjoying. Wama alayna illa al-balagh. Assalamu alaikum wa rahmatullahi wa barakatuh.